Hello and welcome to our new topic. We're going to start this topic by talking about enzymes. Now, we've already talked about enzymes in a very limited capacity. We've talked about them when we were talking about macromolecules because an enzyme is a type of protein. So now we're just going to dive way further into what it means to be an enzyme. To start with, enzymes are catalysts. This is a new word, and this is a word that you should absolutely bold, highlight, put a star next to. You should understand what this term means. So, as I mentioned, enzymes are proteins, but their job is to speed up a chemical reaction. And when something speeds up a chemical reaction, that's called a catalyst. You maybe have heard this term in like English, like the catalyst to the big mid-book, huge fight scene or something was whatever, right? The catalyst to that was this conversation that was had. A catalyst is just something that speeds up a reaction. So for us, in biology, we're going to focus on the idea that enzymes speed up chemical reactions that happen in the body or, in plants or something like that. But they speed up chemical reactions that lend itself to metabolism. So it helps with metabolism, which is, of course, one of our 8 characteristics of life. So this is important in a multitude of ways, but we're going to relate it back to metabolism. Enzymes also have a very specific shape. So enzymes only fit with specific reactants, and we call those reactants substrates. So like this rectangular substrate is the reactant that's going to fit perfectly with this enzyme. Whereas this one, which is like a squiggly glob, this reactant or substrate is only going to fit with this enzyme. But this purple substrate will not be able to fit in this yellow enzyme because they do not share the same specific shape. So we kind of talked about this, but the reactant that the enzyme works on, so the reactant that fits into the enzyme, we call that a substrate. And where they actually combine into one unit, that's called the active site. So wherever the substrate goes into the enzyme and fits nicely, like this little area that's like following where my cursor is, that's called the active site. So when you have a substrate and it's going to come in and it's going to match nicely with an enzyme, and then it's going to create this complex, right, where the substrate is actually connected to the enzyme and you have one solid unit that has been created. The enzyme is going to catalyze whatever it is that is connected to it, right? It's going to catalyze that substrate. In this case, it is breaking down this substrate and then you're releasing 2 products. So it comes in as one substrate, it goes through catalyzation, right? The enzyme works on it, and then you are released 2 products. You can also do this the opposite way, where these 2 products could come in and the enzyme could work on it and put them together, and then the product that would come out would be one solid product instead of 2 separate ones, right? Enzymes can build things or break them down, but the key thing here is that enzymes can be used again and again and again and again. Enzymes are the best thing because they do all of this work, they speed up all of these chemical reactions, they build things, they break things down, and they don't need to be replaced. They just continue existing and doing their job perfectly, and we love them for it. So the jobs, as I mentioned, some break down polymers into monomers. So like in the digestive system, when you eat food, you need to break down like big starches into smaller carbohydrates, and then eventually you're breaking them down into monosaccharides, which then can be used in the body in a multitude of ways. So breaking down polymers to monomers, but alternatively, enzymes can also build polymers by combining monomers. And that's like when you build muscles, you're putting amino acids together to build more protein, which helps you bulk up and get more muscles. Some examples of enzymes. Typically an enzyme is going to end with the suffix ase or in, and you'll see that I underline them in these examples. So amylase, right, ends in ASE, that breaks down carbohydrates. Lipase breaks down lipids. And that one kind of makes sense, right? Because like LIP at the beginning is the same as the LIP in lipids. Protease breaks down proteins. Again, similar beginnings of the word, so easy to remember. And then pepsin breaks down amino acids in proteins. So that's like, once protease breaks down proteins, then pepsin breaks down those proteins into even smaller parts. So the only one that's like super out there is amylase, because amyl, the beginning of that word, doesn't really go well with carbohydrates. So that's just something you're going to have to memorize for this one. But the other ones sort of go hand in hand with what they break down. And now the last thing we're going to talk about is how enzymes can denature. When an enzyme is in a bad condition, it's going to denature or basically break. And the two conditions that we're going to focus on is temperature or pH. We like to have our body in a very consistent temperature, right? If we go too hot, we have a fever, right? We start sweating, we feel sick. It's not good. Well, it's not good for our enzymes either. And if our body gets too hot, our enzymes are going to denature, and you can see that right here. If our temperature goes above like our normal body temperature, ooh, the enzyme denatures, it breaks, and all of a sudden it doesn't work well. And the same thing can be said for pH. Remember that pH is how acidic a substance is. The lower the pH, the more acidic. The higher the pH, the more basic it is. But enzymes also have an optimum or ideal pH that they work in. And if they go too far acidic or too far basic, you're going to have the same problem where the enzyme is going to denature and you're going to lose enzyme activity. And just to really, bring home this point, to denature an enzyme means that the enzyme loses its shape. When it loses its shape, that means it does not have an active site. And if it doesn't have an active site, that means it can't match up with a substrate. And if it can't match up with a substrate, it can't perform its chemical reaction. It's useless to us at that point. So a denatured enzyme is like the worst thing that could happen, because that means all of the chemical reactions that it's normally performing are not being performed anymore, and it's making our bodies run at a slower speed because we're not able to actually do any of these chemical reactions at the pace that we've been doing them. And so we want to stay in a consistent temperature and a consistent pH, not just in our bodies, but for every organism that uses enzymes. They want to be consistent, right? It's all about maintaining homeostasis to be able to do all the metabolic chemical reactions that we need to do to survive. Okay, great job. I'll see you in class.